Hello, Assalamualaikum. My name is Amar. So today I want to talk about DNA duplication, RNA transcription, and protein transcription. Okay, so we start with DNA duplication. So DNA duplication happening when the process DNA molecule made an exact copy of itself. And this is happening before the cell divides, so itself can create a new cell for receive the same genetic information as original cell. So the step of DNA duplication happening is like unwinding, replication, joining, and the result at the end. So the first step of the DNA duplication is the unwinding, which means the enzyme helicase unzip the double-stranded DNA and break the hydrogen bonds between the base pair AT and the GC. And the second one is replication. So the enzyme DNA polymerase adds the new nucleotides that makes the original strains, which means A pairs with T and G pairs with the C. So the next is joining. The enzyme ligase connects the newly made DNA fragments, especially on the lagging strands, into the continuous strain. So the result is two identical DNA molecules are formed. So they each contains one original parent strain and one new strain. This is called a semi-constitutive replication. So the second one is RNA transcription. So RNA transcription is a process where a section of the DNA is a copy into the RNA. So this is the first step making protein from a genetic information. So RNA transcription is step by step. Start with initiation, and then the unwinding. The next one is elongation, which means it's building. So start it with initiation. So the enzyme RNA polymerase binds to a specific region on the DNA called promoter. So this mark transcription begins, and then it continues with the unwinding. So the DNA double helix will unwind, and one strand of DNA acts as a template for making RNA. So the next step is elongation, which means building. RNA polymerase adds RNA nucleotides such as A, U, C, G to form a single strand of messenger RNA, which means mRNA. So in RNA, the uracil, which means a U, is replaced by a thymine, which means is a T, and paired with adenine, which means A. So the lastly is protein transcription. So the protein transcription is the first step of the protein synthesis, where the genetic information from DNA gene is copied into the messenger RNA, which means mRNA molecules. This process will occur in the nucleus and carry out the enzyme RNA polymerase, which use the DNA as a template to create a complementary mRNA strain.